We're now going to start to talk about the process of mitosis and cytokinesis. In other words, actual cell division. First, mitosis, and the first stage of mitosis is prophase. Prophase is pictured here with a plant cell in prophase at the top and an animal cell in prophase at the bottom. Here's our representation of prophase, and you can see some key things going on. Number one, you can see that the nucleus, the nuclear membrane specifically, has started to disintegrate. In the midst of that nucleus, you can see that the chromosomes here, and it pictures 4 dyad chromosomes, those chromosomes are now visible. In addition, the centrioles, a pair to the right and a pair to the left, are moving in opposite directions and ultimately will end up at far ends of the cell, here and here, which we would refer to as the poles. Fourth and final is that those centrioles are producing these thread-like structures that you see in the picture called spindle fibers. It's important to note as we talk about prophase that number one, you could see the chromosomes. Well, there's a reason for that, and it's not just that the nucleus was disintegrating. If you look at this picture and look down at the very bottom, you'll see a double helical shape of DNA in dark blue and light blue. Just above that, you will see that DNA right here is wrapping around these yellow protein structures referred to as histones. And then those DNA and histone combinations are then wrapping around each other very tightly, which gets you to this structure that you see here sort of in the middle. This is the normal state of your genetic material. When a cell is in interphase and the cell is doing the job of that cell, this is how your DNA exists in this sort of level of sophistication. And that is referred to as chromatin. Chromatin is so diffuse, it's so spread out and thin that you couldn't see it. Even if the nuclear membrane were gone, you wouldn't see this material. You can see this material, however, during prophase. Why? Because the chromatin then bends and folds, as you're seeing here, and then wraps itself around all the other DNA and coils. And ultimately, if you look finally at the structure here at the very tippy top, there is so much DNA wrapped and coiled and surrounding itself that you get lots of it in the same space. It's dense enough that you can finally see this level of sophistication in a microscope. You see a chromosome, and that's why you can see the chromosomes during the prophase, because this is the form that DNA occupies during that time. So chromatin normally, but chromosome during the process of cell division. From prophase, we move on to metaphase. Plant cell metaphase pictured here at the top, animal cell pictured here at the bottom, and here is our representation. Now the key always in mitosis is what's happening to the chromosomes? Where are they and what are they doing? And in this case, the chromosomes are lined up all on the middle of the cell as pictured right here. You can see that the middle area of the cell in this picture is labeled as the metaphase plate. It's sometimes referred to as the equatorial plate. It's most commonly just referred to as the equator. That's number one. That's key to metaphase. Where are the chromosomes? They're on the equator. The other key factor that you have to take notice of here, though, would be the spindle fibers. If we focus in on this chromosome at the top, the orange-colored one, you can see that it is attached by a spindle fiber from the left-hand side, but it is also attached by a spindle fiber from the right-hand side. That's key in setting up what's about to happen. Every chromosome is attached twice. What happens is anaphase. Plant cell anaphase at the top, animal cell anaphase at the bottom, and of course our representation here. And basically you can think about anaphase as being a game of tug of war. The spindle fibers are shortening, In other words, they're pulling in opposite directions. And so again, if we look at what was that one chromosome up here at the top of our cell, you can see that it has been ripped in two. There is one chromatid heading towards the right, and there is another chromatid heading towards the left. But remember, they are exact replicas of each other. That gets us to telophase. Now, telophase is unique in that, yes, it is the last stage of mitosis, but if you remember from an earlier lecture, cell division as a whole is made up of two main parts. There is mitosis, which is what we're talking about now, and there is cytokinesis, which divides up everything in the cell other than the chromosomes. The odd thing is that both of them can happen at the same time. So yes, we're talking about telophase, the last stage of mitosis, but at the exact same time, we also have to talk about cytokinesis because they're both occurring now. Here would be a plant cell pictured in telophase, and down at the bottom again, an animal cell. Our representation here shows you the one simple thing that you need to know about telophase. If you look here on the right side, you can see that a nuclear membrane has formed around the genetic material, and the exact same thing has happened over here on the left side. When we started this whole process, there was one nucleus that disintegrated, and now you have two nuclei reforming. That's it. That's all of telophase. But of course, that's not all that is going on here because cytokinesis is happening at the same time. If we think about cytokinesis in an animal cell, we can look at this picture in the lower right-hand corner. You can see that there is an indentation occurring here at the top, and there's another indentation occurring here at the bottom. Those indentations are referred to as cleavage furrows, and those cleavage furrows will just enlarge. That indentation will get larger and larger, and what is currently just one cell will ultimately split into two. Now, that's the way it works for an animal cell, but it can't be the way it works for a plant cell. Why? Because a plant cell has a cell wall, a tough, thick, rigid cell wall that would not indent like an animal cell does. And so if we look over here at this picture to the left, you can see that we have one cell. Now, we have two nuclei. There's one on the left, there's one on the right. And in between those two, you have forming what is referred to as a cell plate. And a cell plate is nothing more than the beginnings of a new cell wall. This cell plate will get bigger and thicker and larger, and ultimately it will become a cell wall, in which time this one cell won't be one cell anymore, it will be two. And cytokinesis will be complete for the for the plant cell process. This would probably be an appropriate time to also mention that there is one other distinct difference between a plant cell and an animal cell going through cell division. I've been mentioning throughout the idea of the centrioles, but of course centrioles are only present in animal cells, not in plant cells. That brings us to one final idea when it comes to cell division, the fact that there are chemicals within the cells called cyclones that control the pace at which cells go through this whole cell division process, the whole cell cycle. You can see that there is a variety of different types of cyclones here in this picture labeled cyclones A, B, D, and E, and that they rise and fall at different points in the cell cycle and essentially push cells through that cycle.